A warm good afternoon to all gathered here for this session on applications of high performance thin layer chromatography in several areas of biology as well as other fields. It's at the outset, it's my privilege and pleasure to accord a warm welcome to our Secretary and Director, Dr. C.A. Vasiki. Madam is very proactive. She is a great scientist and a very good administrator, and she is giving a lot of support to such activities in our campus. She is currently in London with her son, and it's with great sense of honor I welcome Madam for the session. I welcome our most dynamic principal, Dr. M. Lakshmanaswamy sir. Sir is actually our teacher, and he was the first HOD of this department of Biotechnology department. And I am a proud alumni of this department as well. I welcome you, sir. I welcome the HOD of Biotechnology undergraduate program, Dr. R. Sivaprakash, all the faculty members from the Department of Biotechnology and students who have joined here for this program. It's my very privileged and proud state to welcome Dr. Saikat Malik, Dr. Amit Palande, and Mr. Rahul, the team from Anchrom Technologies, who have graciously accepted our invitation to be a part of our venture in providing insights about industry, in providing insights about what are the actual requirements of the industry to our students. On behalf of our management, principal, and all the faculty members, I once again welcome all of you for this program. Thank you so much, and hope we will have a wonderful session in the next hour and so. I now request our principal, Dr. M. Lakshmanaswamy, to speak a few words about the program, and then we will start the session. Thank you.
Good afternoon, all of you. Most respected secretary and syndicate member of Bharatiya University, Dr. C.A. Vasiki Madam. Our guest today, Mr. Amit Palande, Application Chemist, Mumbai. Dr. S. Madan Shankar, Dean Academics and Head PG and Research Department of Biotechnology. Dr. R. Sivaprakash, UG, HOD of Biotechnology. Guest lecturer, PG and Research Department of Biotechnology. Ms. S. Bhargavi, faculty members, participants. On behalf of the management, welcome you all for Industry Academia Lecture Series Talk 2 on Hptlc technique and applications with a live demo. Anchrom Hptlc specialist since 1978. High performance thin layer chromatography or Hptlc is one of the sophisticated instrumental techniques based on the full capabilities of thin layer chromatography. The advantages of automation, scanning, full optimization, selective detection principle, minimum sample preparation, hyphenation, and so on enable it to be a powerful analytical tool for chromatographic information of complex mixtures of pharmaceuticals, natural products, clinical samples, foodstuffs, and so on. Its main key features are simultaneous processing of the sample and standard better analytical precision and accuracy, less need for internal standard. Several analysts work simultaneously, lower analysis time and less cost per analysis, low maintenance cost, simple sample preparation, handle samples for divergent nature, no prior treatment for solvent like filtration and degassing, low mobile phase consumption per sample, no interference from previous analysis, fresh stationary and mobile phases for each analysis, no contamination, visual detection possible open system. Application of Hptlc for phytochemical analysis, biomedical analysis, herbal drug quantification, analytical analysis, fingerprint analysis, and Hptlc future to combinatorial approach, Hptlc MS, Hptlc FTIR, and Hptlc scanning, diode laser, and Hptlc power analytical tool in the field of analysis. It is noteworthy that utilization of instrumental Hptlc towards the analysis of drug formulations, bulk drugs, natural products, clinical samples, foodstuffs, environmental and other relevant samples will increase in the future. I am also eagerly waiting for our chief guest demonstration. Thank you for the opportunity given to me. I wish the program a grand success. Thank you.
Thank you, sir. Dr. Amit and Dr. Saikat, the floor is yours and you can start the session.
Okay, sir. Thank you so much for
Thank
wonderful
you so
introduction,
much.
sir. Yeah. So, good afternoon, all. My name is Amit. I'm working as an application chemist in Anchrom laboratory, which is situated in the Mumbai. We are really grateful to organize this webinar with the KONGUNADU Arts and Science College. Hope so. This session will be very useful to PG students and everyone. So now I will start my presentation. So I will share my screen, and during the presentation, if you have any queries, doubts, you can ask us in the QA section. There is my colleague Ramakan and my sir, Saikat sir was there to clarify your doubts. So my screen is visible to everyone.
Yes, Amit, yes, it is visible.
Okay, thank you, Ramakan. Okay, so first of all, our topic for this presentation webinar is the high performance thin layer chromatography technique and its application. In the short form of this is the HPTLC, high performance thin layer chromatography. It is not HPLC, just like where in HPLC P stands for the pressure, but here in the HPTLC P stands for the performance. Now, our these are our social media contacts. If you want to follow us or have any doubts or queries, or if you have any questions, you can contact us on this social media. First is a LinkedIn, then the Facebook, we have official page. Then if you go on the YouTube, and there is one official website is the www.anchrom.in. So you can contact us on these social media accounts. uh, if you want to have any queries, doubts, so you can reach us on this WhatsApp mobile number 7506792122, or else you can send us whatever your query, doubts, or anything information, lab@ancrom.in email ID. Now, this presentation is specially designed for the Zoom platform or the Google Meet and Microsoft Teams. I always feel proud because our Anchrom team is giving the free HPTLC training from the 1994. So, and it's a free training for the all government employees, PG students, then their teachers in our HPTLC research lab, which is situated in the Mumbai. Or else training can also be arranged in the all cities, and for that the dates will be decided mutual convenience. And after the each training, we issue the certificates for that training. Till that, over 5,000 Indian scientists was trained from 1994. And we have the MOUs with the several prestigious universities and institutes. This is our Anchrom YouTube channel. If you go on the YouTube and just search the Anchrom, you will get the lots of video related about the HPTLC. So there is one example, is a training videos for both Weed Cats and Vision Cats, and several professors are using our videos in their teaching courses also. So very useful. Now, first, what is chromatography? Chromatography means what? It is the technique of separation. Means if you're having any mixture, you want to distribute these components between the stationary phase and mobile phase. So it is a technique of separation of mixtures. So here we use as a stationary phase is the our silica plates, which is not movable, and the mobile phase in which we will dip our plate to separate that components. Now, evolution of HPTLC. The TLC started in the 1960s. After that, some years after, instrumental TLC came in the market. After that, software controlled TLC came. Then GLP compliant TLC came. Like that, HPTLC methodology, then pharmacopoeia HPTLC, and then at the end, regulatory compliant HPTLC in the market. And now in the 2021, fully automated robotic HPTLC launched by the CAMAT. Now, here you can see the TLC versus HPTLC comparison. So at the right hand side, you can see at the yellow plates, which is the analysis of Asian ginseng, which is our traditional method. Take a capillary, do the spotting, run the plate in the this suitable solvent, then you get the separations. But at the left hand side, you can see there is a rectangular plate, which is the HPLC plates, and there we don't do the spot application. There we go for the band application. So you can see the difference between the both separations of analysis. So in the ginseng, at the right hand side, very poor separation, which is very clumsy, not clear that much. But if you see in this plate on the HPTLC 20 by 10 plate, there is a very good separation from the baseline. And at a time you can compare with the 4 to 5 standards. So you can see there is one pure standard was a bands applied, and you can compare it with the sample. And at a time you can do the 15 samples on 20 by 10 plates. It is not in the normal our TLC plates. So these are the different. It was just like that. समझो हम कुछ साल पहले नोकिया का फोन्स यूज करते थे, लेकिन after some times in the market Samsung phone come with the Android features. That means it giving the some updated features. So it is just like that only. TLC, its updated version is the HPTLC. Now here you can see also see the difference. Most of the monograph says that use a TLC plate 20 by 20 size, and HPTLC always go for the 20 by 10 plate. You can see in the TLC plate we have to run this plate up to the 135 mm of the solvent front. But in the 20 in HPTLC we run only the 62 mm. That means the solvent runtime will be reduced, the solvent used for this development will be reduced, and the separation, here you can see the separation in the 20 by 20 plate, the separation was somehow blurrish, not that much of clear and the good. But here you can see in the HPTLC the same separation, we are getting it very sharp bands. So miniaturization, we called as a miniaturization. So very less solvent, less time, and the sharper bands you will get in the HPTLC. So this is the example of TLC plates versus HPTLC. Now here you can see the TLC versus HPTLC comparisons. First of all, both are the planar chromatography. Primary focus in the TLC was the very simplest and low cost, but in the HPTLC you go, you will get the reproducible results. Means if today you are doing one analysis and the same analysis you will do the tomorrow or the one after the 1 week, you will get the same reproducible results. It is not like in TLC. In the TLC, we are doing by the capillary. Today I am doing, I don't know how much volume it is apply onto the plate. Tomorrow also I am going, so it will not clear. Today it will apply the 2 ml. Tomorrow it may be 2.2, 2.5 ml, but it is not in like the HPTLC. HPTLC, you know how much of the volume you are applying onto the plate, and the separation power which are getting in the HPTLC, we already seen in the slide how much good separation we are getting in the HPTLC. Is a process flexible, no rules there in the TLC, but in the HPTLC there are the standardized methodology, optimized parameters was there. Methods wise, you can see there are very few parameters were defined in the TLC, but in the HPTLC well defined parameters with the validation. Flexibility very high, and in HPTLC none for validated methods, high between the methods. Targets, you can see in the TLC you will get the rapid result, preliminary result, but in the HPTLC you will get the reliable analytical results. Then uh, cost for this analysis is very low in the TLC, and in the HPTLC you can see low per the sample as compared to the HPLC. Plate size in the TLC you can use any format, but most of in HPTLC 20 by 10 or 10 by 10 plates. So these are the basic difference between the TLC and HPTLC. Here you can see the updated HPTLC module systems and data output. So first of all, in we do application by using these applicators. Now we will not use the our manual technique, which is capillary we are using for application, but now we will use the applicator. So first applicator was ATS4, which is fully automatic TLC sampler. After the application, we'll go for the development part. So for development, we have the ADC2, which is automatic developing chamber. After that, we go for the photo documentation part. For that, we have the visualizer. After visualization, we'll go for the scanning data, where we will get the RF and area. So scanner we will use. After that, if you want to find out the M by Z ratio, mass to charge ratio, or for any unknown compounds, you can go for the MS interface hyphenation technique. And in the end, there is a derivatizer, and all these modules of HPTLC are connected with the one software, which is VisionCat software. Now here you can see the HPTLC quantification. Uh, so first of all, standardized methodology is there. After that, validated methods. Key methods are validated, so you will get that specific results. Software controlled hardware. I already told you these all modules are connected and controlled by the one software, which is VisionCat. After that, sensitivity of this instrument is till you can go in nanogram in UV light and at the picogram in the fluorescence light. Then there is a reproducibility plus minus 2%. Accuracy and precision is very high. You can do the multiple detection without repeating that chromatography. MS hyphenation technique was there, and fully regulatory compliant worldwide. HPTLC is a very simple, reliable, fast and safe technique. You can see there is the simplest and fastest chromatography method. Ultra low running cost and maintenance. It was not like that in the HPLC. You have to on the AC for 24 hours. In the HPTLC, it is not necessary that you want AC 24 hours on. Very rugged and very powerful instruments of HPLC. Multiple detectors are there. Normal eyes we can see, but there are the electronic devices just like visualizers and scanner. And after finish analysis, you will get the UV data. You will get the data in the UV, you will get the data in the visible light, get fluorescence, you will get the spectrum, derivatization data, and the MS data. So huge data you will get for your samples. Now HPTLC fingerprinting. Fingerprint means what? There is a unique identity of each one of we having. I have unique prints, other one having, everyone having their unique fingerprints. Just like that in HPTLCS2, each and every sample having their unique identity. Okay, so we called as a fingerprint. Means after the separation, I know that these are the specific marker or the main components will be present. Just like suppose in the turmeric powder, I know curcuminoids. There is a bismethoxy, dismethoxy, curcuminoids are there. So after if I saw specific main components in the present in that sample, so I can definitely say that this is a turmeric powder sample. It just like that unique identity. So you will get is the image with visual HPTLC chromatogram based on the RF color and intensity of the bands in the electronic images. Then you can compare it with the BRM. BRM is botanical reference material and can be stack multiple images in different detection modes like 254, 366 and white light. So here are images of the printings. So see, this is the example of HPTLC fingerprinting is unique. So here I took the different samples, the Piper nagrams having this unique identity, then Piper longum, then Piper retrofractum, Piper pubeba, Piper borbazens, and xanthosylum. So here you can see each one of them having a unique identity. So here you can see if this is xanthosylum piperatum, this band is not in the Piper nigram or Piper longum. So this is something marker compound in this present in this. So on this basis, you can after it is manually you have to take out, but here it will automatically come out from that chamber in the upper part and automatically it will be dried. So this ADC2 is the USP compliant, software controlled, fully automated and reproducible result. Main feature and main benefit of this instrument is what? It will provide you the humidity control. Means there is one jar with this instrument that containing the magnesium chloride hexahydrate solution, which will provide you the 33% of the relative humidity. Then automatic monitoring to the solvent front was there, automatic drying to the plate. So this is the benefit of this ADC tool. Now this is the 2D chromatography. So if you wanted to find out the stability of your sample, so you can do the 2D chromatography. At the one corner you will apply your sample, you run that sample into the solvent. After that you just turn to the 90-degree and again develop with the same solvent or the another solvent. If you find out there is another fractions like this in the second, there is a fractions you can see, so that means there is some degradation is happening to the sample, so this is unstable. And if you find out the no fractions in a one line separation, so that means your sample is the stable. So this technique is very useful to find out the stability onto the silica gel bed. So investigation of stability during the chromatography mixtures 1 and 2, same mobile phase you can use. Now this is the gradient development technique, which is the AMD2. Now AMD2 means automatic multiple development chamber. So what happened is in the AMD2, here you can see there is a 5 bottles. Mostly these modules is used for the R&D purpose or for the any method development. So in these 5 bottles, there is a different solvents filled with the polar to mid polar, mid polar to polar and non polar. So like that. And here you can do the multiple development up to the 25 steps in a same direction with one plate. I mean, suppose if you are applying your sample, then on the first 20 you have to develop in the only methanol. After that 20 to 30 you want to develop in the toluene. Then 30 to 50 you want to develop in the another hexane solvent. So just like that you can do up to the 25 times development by using this AMD2 with the 5 solvents. This is the example of camera application node, phospholipids node, and there is one CBS. Now, after the development, we go for the photo documentation part. So, for photo documentation part, we use this CAMAC TLC visualizer. Now, this CAMAC TLC visualizer will give you three types of images. First is the white light, then 254 for the UV, and 366 is the fluorescent. So, you will get the three types of photos, and these photos are very HD photos because there is one high definition CCD industrial camera was fitted here, which is specifically designed for this module. You can do the comparison study by using this. So this is the example of curcuma. So this all data is collected in the different sample days, lab species and analysis, and all this data collected in the one comparison viewer, and you can see the data. So from that you can compare it, any degradation was happened or anything extra band or anything extra was present in the your sample or not. So this is the example. So this is the comparison you can do by using the PLC. Now after this photo documentation, the next important part is the scanning part. Scanning means by using the densitometry scanner 4, you can go for the quantitative analysis. This is the heart of Hptlc. So this scanner range is the 190 nanometer to 900 nanometer. So it will cover the UV, visible and the fluorescence also. Fully automatic. So you will get the scanning data. You can go for the spectrum analysis, and it is very sensitive and reproducible. Why it is sensitive, I will explain in the later slide. And the data which we are getting is the non-destructive measurements. So here you can see the example. Now the chromatographic evaluation. So this is our plate at point RF point 1. I am getting this band. When I will scan this band under the scan by using the scanner, I will get the peak like this, very sharp. Then absorbance spectra. Why we are taking the spectra? Because when we go for any specific band spectra, that will give you the lambda max value, and depends on that lambda max value you can find out which is this sample or which is this compound. So here you can see for specific we have sulfa methoxyl 260 nanometer, trimethoprime 210 nanometer, and dimethoxyl 318 nanometer. So 210 is a very low range UV. So this can will also give you the response at a lower UV range also. So this is the benefits, uses of absorbance spectra. When you take only one spectra, that gives you the identity. But when you take the spectra 3 to 4 times, that will give you the purity of that spectra. Then at a time you can do the multi-wavelength scanning also. So here you can see at a 205, 268 and 311. At a time I will scan my plate with the multiple wavelength. So this is the benefits of this scanner. Quantification of the Hptlc scanner for the cannabinoid. So this is the example. So here you can see the TSC means tetrahydrocannabinoids is not visible in the 254 after the development. Okay, and this band is for the THCA means tetrahydrocannabinoic acid. But after the scanning, I can say that there is one band which is there. This line was drawn here. So at a 210 nanometer, it is detected. That is at a lower rate, but it is not visible at a 254 nanometer. But scanner, it is detected. It's showing the peak for that. So here you can see this is a peak, and for the tetrahydrocannabinoids, it is showing the 285 nanometer. So this is the use of the TLC scanner. After the scanning, we'll go for the derivatization. Now, most of you want to know what is derivatization. So derivatization is the process to giving the colors to the bands. What happened after the application and development? I didn't observe anything under the UV or the white light or in the fluorescence. So that time you need the derivatization. Means with any specific reagent, you will react that plate. You will or either spray that plate, and then you will the bands will be showing the colors. So that process we call as a derivatization, giving chromophores to the colors. That is a very unique technique in HPTLC and applicable to every sample. So you just need to spray with reagent and heat that plate. Get additional info about that. For heating, you can use this Canac plate heater. So here you can see this is the plate heater. You will get the very additional information about that sample, which is not available after development. But after derivatization, you will get a very additional you can go for specificity. Example, detect functional group or class of compound. Very ultra high sensitivity is there, and visual confirmation, which is rarely required if scanner, because after the giving colors it is visible to our eyes. So there is no early scanner is required for the scanning procedure. So for derivatization, you can use this Camac derivatizer. It is the pattern technique. The it is works on the piezoelectric effect. So here you can see this is the nozzles. This is the blue color nozzle. Like this, there are three more nozzles: yellow, red, and green. And this is blue. So four types of nozzles was there. What we have to do? This is the hood. Inside this hood, you have to keep your plate. Fill this nozzle with your reagent. Now, here you require only 3 ml of the reagent for 20 by 10 plate. And you can imagine only 3 ml of the reagent is sufficient for analysis. So how much volume you are saving? So after the pressing this start button, it will start on spraying that reagent onto your plate in the form of mist. After that, mist will be settled down in your plate, and the vapors were generated, which is extracted, means evacuated by using, there is a vacuum. So beside there is one bottle which was containing ethanol and water mixture, and that all vapors will be suctioned onto that bottle. So there is no, I can say that there is no contamination or the like pollutions. The vapors will not come out from this hood. So very green, eco-friendly, eco-environmental friendly, we can say this technique. That is why this technique is called as a green technique. So here you can see some of the examples of the derivatization process. So after derivatize with different reagents, you can see how these color bands will be seen. So there are 14 different classes of compounds: alkaloid, anthracene, beta principal, coumarins, essential oil, tannins, sterols, affinities. So these all bands are giving colors. Some bands giving yellow color, violet, pinkish, reddish, bluish. So after the derivatize with the specific derivatizing reagent, you can see this type of color bands. Now HPTLC MS interface hyphenation technique. Earlier told you, if you wanted to find out any unknown compounds to M by Z ratio, mass to charge ratio, so you can go for this technique. So it is you just have to hyphenate with the pump and the MS. So confirmation and characterization, you chemical synthesis, phytopharma, pharma and R&D, foods, it is used. suitable for any MS and MSMS and HRMS analysis. Now, how it works? So the left, you are keeping a hyphenation technique at the center. At the left hand side, it's connected with the pump, and right hand side connected with the MS. So the outlet of the pump is connected to the inlet of this interface technique, and outlet of this interface technique is connected to the inlet of the MS. So the mobile phase come from the HPLC pump. It will go with for specific that bands, and it will dissolve and it will transfer to the MS, and you will get the in a 10 second you will get the result like this. So the very simple to use in a 3 minute cycle cycle for MS analysis. Only pure fraction eluted, very cost effective MS analysis, because up to the 100 MS per day you can do for selected fractions. So it increase the existence of LCMS or LCMS productivity of any make in 10 times. Here you can see this is the USP chapter for 2251 for the adulteration of dietary supplements with drugs and drug analogs by Hptlc MS. So this is the one example. So identification of API. API is the tadalafil in Ayurvedic sample. We all know tadalafil is a synthetic API, active pharmaceutical ingredient, which will not present in the Ayurvedic sample, but it is present into the Ayurvedic sample, and it was detected, and then it was identified by using the MS hydrogen technique. This is the Shimadzu Anchrom joint poster, which was one collaboration event was held in the USA. ASMS conference was there. So this is one example. Now this is now another module, Kamang bioluminescence. So Kamang bioluminescence is a bioactivity detector via bioluminescence. Now I will tell you bioluminescer is means what? By using this, we can detect the toxicity of the sample by using this bioluminescer. If you wanted to find out the toxicity, and it is not directly related to the HPTLC, but the initial step was the same. You have to apply the sample, you have to develop your plate, and after the development, you have to dip the plate into the solution of the bacteria. Mostly we use the solution of the bacteria is the alivibrio fishery. Now why only the alivibrio fishery we are using? Because very safe to handle that bacteria. Then when there is a very healthy colony of bacteria, they will emit the fluorescence, that means the bioluminescence. They are extremely sensitive to the uh, even very minute level of the toxicity, and they show the dark and blackish color of band. So here you can see this blackish color of band showing there the toxicity of in the samples. So by using this bioluminizer, so this under this bioluminizer you have to take the photos, and you will get the photo like this, and it is showing this black color bands, that is the indicate the dead bacteria. So you can say that there is something bacteria was present and which was dead by using this alivibrio fishery. So this is the example of the fingerprint of wastewater sample. So by using this luminizer you can find out the toxicity in your sample. So selective detection of biological activity in the synthetic mixtures or the natural products, then toxicity of profiling of water, then toxic residues and material detections in the food, beverage, dietary supplements you can do. Toxic residues in the soils you can find out by using this bioluminizer. Now the HPTLC methods and validation, very simple and rapid technique, needs very few plates and 1 day. Then validation takes 3 to 5 days. New method development and validation cost only 25 to 30,000 rupees. For many new method development and validation also. So very cheap if we compare to the other technique. Possible to create and validate 10 to 15 methods per month on HPTLC. Now, these are the HPTLC method validation according to the ICH guidelines. They decided these parameters should be carried out to do validation. First is the specificity, then precision, intraday and interday, accuracy, linearity, then LOD, limit of detection, LOQ, limit of quantification, and ruggedness. Now, now we see the HPTLC. Now that's HPTLC updated in the HPTLC Pro, which is the fully automated. So this is the full system HPTLC Pro. So this system is interconnected with each other. Earlier HPTLC system having the each different module, having one each everyone having each different model, but this HPTLC Pro is connected with each other. So you can see first is the applicator, the development, derivatization, detection, and interface. It is connected. So there is inside there is a conveyor belt. So it will transfer that plate after application to development, development to then identification, then derivatization, scanning. So it is interconnected. So I will show you the small video which will give you the idea how it is works, the actual video. So here you can see the two modules, applicator and the development. So by watching this video, you will get the idea how it works, the HPTLC Pro. Now the HPTLC application areas. Where is the HPTLC is using? So HPTLC first pharmaceuticals, food and adulteration, botanicals, industrial analysis, reverse engineering, organic chemicals, reaction mixture, and forexing. These all fields are using the HPTLC system. First, we will see some examples of the HPTLC in the pharmaceutical application. So here we can see first example of quantification of the glimepiride tablet in 1 mg tablets. Okay, so we all know that glimepiride tablets we are using as anti-diabetic tablets. So in the UV light it is visible. So here you can see the image. After the scanning we plot the graph and we quantify that glimepiride tablets content. So here you can see the 133.3 mg is present in the 10 mg of the tablet. So by using the HPTLC, you can do this. Second example is the quantification of the afexabine in afexabine tablets. Now we all know the afexabine is an anticoagulant tablets that is used for the to prevent the blood clots or the prevent the strokes in the people. Used as a blood thinner. So this is visible in under fluorescence we can see, and after the plotting the graph 96.25 μg is present in the 100 mg of the tablets. Another example was the miltefosine tablet, which is the antimicrobial drug, and after the derivatization, it requires the derivatization because after development it is not visible, but after derivatization you can see there is a color bands which is observed here, and after the plotting graph you can graph, or you can do the after development, or after the uh, plotting graph, you will get the result. Then degradation of profiling of cefixime and azithromycin. Now degradation study you can do by using Hptlc. Now these two drugs are antibiotics drugs, and for this analysis, this publication means the Vandana Gawande ma'am did, and this publication was international award in Euro 2000 at Berlin. Okay, so here you can see the cefixime degradation. So they did the degradation study in the different conditions for the acidic, basic, oxidation, heat, and photo. So in the heat, in cefixime showing the degradation, and for azithromycin in the acidic condition it is showing the degradation. So by using this HPTLC, beneficial is for stability testing, and it is disposable stationary phase, as well as in case of the forced degradation when appreciable amounts of the acidic, alkaline, and peroxide reagents are used. So you can do the degradation study by using this HPTLC technique. Then the simultaneous RP-HPTLC methods for quantification of levodopa, carbidopa, and entacapone in the fixed drug combination. So if any drug having the fixed drugs combination, so that fixed drug combination you can use, or you can quantify or identify by using the HPTLC system. So the fixed drug combinations are non-regulated by pharmacopoeia, and therefore ideal for the HPTLC analysis, which is 10 times cheaper as well as the faster than LC or any FDC phase. Fixed drug combination analysis methods can be validated. Now, HPTLC method for estimation of ulipristal acetate in the this presence of the its forced degradation products. Now we all know this ulipristal. Ulipristals is as an emergency contraceptive or the uterine fibroids treatment. We use that medicine, and by using this HPTLC technique, you can go for this forced degradation study also. Then the HPTLC determination of diclofenac sodium from serum sample. So diclofenac is a painkiller, we all know that, and this method is very sensitive and reliable. By using this HPTLC technique, we can identify or determine the diclofenac sodium from serum sample. And if we go further studies with MS, this is also possible. Cleaning validation of HPTLC. So by using this HPTLC, you can do the cleaning validation also, because suppose in the pharma sectors, they completed the one batch. After that completion, they clean their chamber and vessels, and they will go for the further batch, next batch. But before going that further batch, they will analyze their chambers and the vessels are clean or not, for they will take the swab and like that, and that analysis you can do by the HPTLC. So this is the cleaning validation. If they observed anything, bands or fractions, so you can say this is not clean. Or if there is no any fractions or the extra bands was present, so you can say that it is clean. That so comparable results between the HPTLC and LC procedures. One second. Then evaluation of HPTLC chromatogram by densitometry, low detection limits, side by side analysis with many samples you can do. Then fast identification of unexpected impurity during the API production by HPTLC MS. So sometimes what happened during the process of control, there is any unexpected impurity was detected by HPTLC, and whole production process was stopped, and for identification of that unknown substance. So this is very beneficial technique uh, during the production. And further, for that unknown impurity, you can go for the MS analysis also. Then content uniformity test. So by using this HPTLC, you can go for the content uniformity test. It is very ideal test by using Hptlc because it is very fast and low cost. A typical cut cost is around 1500 by Hptlc, and it almost cut up to 8% can be done per day by day. After this, some botanical examples were there. First is the identification of the Vitis nigundo, which is the chastity leaf by Hptlc fingerprinting. So method can be used for identification of Vitis nigundo with markers. So there are two main markers, which is the agunoside and nigundoside. So they took the different species uh, from the Chinese and the markers different regions, and then they analysis and identify. Next is the identification of Ashwagandha, Withania somnifera. We know that there is a two main components, withaniolide and A and beta cetosterol. So by using this Hptlc, you can go for identification of this Withania somnifera. So for that they took the different roots of Withania somnifera from different regions, and then they compare with the beta cetosterol and withaniolide A, and then they compare the and identify the study. Then identification of Kokum, Garcinia indica, fruits by Hptlc. So Garcinia indica consists of the dried pericarp of the fruits. So there is they took three different species: Garcinia indica powder, Garcinia gummigatta, and Garcinia ex mangostana. So they did the application after the development. They observed this. Then identification of nimesulide and diclofenac in Ayurvedic tablets. As we know, nimesulide and diclofenac is the API, which is synthetic APIs, which should be not present in the Ayurvedic tablet, but sometimes they add it in the Ayurvedic tablets to give fast result. So if that is present in the Ayurvedic tablets, so this is not permissible. Uh, so you can see that we can do identification of this APIs by using the Hptlc. Quantification of adulterant, C. xanthorosia or C. aromatica into the sample. So this is the curcuma longa sample. If there is any adulterant was added, so you can do this adulterant study by using the Hptlc. Steroids in the herbal formulation. If steroids are present in any herbal formulation, that also you can identify. So this is the example of prednisolone, hydrocortisone. We can see by using the Hptlc, you can go for the stability study in the Ayurvedic drug also. So for that, you can do by the quantification also, or by the fingerprinting method also. Both method you can do. So here is the quantification for curcumin, right? In the 2018, it was showing 0.986%. But in the 2019, that same sample is showing 0.924. That means something was degraded or extra uh, happen in the sample, so degrade that sample, so it is which was not stable. So by using this, you can do the stability study also. Some of the examples in the forensics and food analysis. So USP chapter 202. Now this is the very official chapter for the fixed oils, for like flaxseed oil, canola oil, or the corn oil, peanut oil, soybean, sunflower, sesame oil. So each one of them having their unique fingerprinting, as I told you, unique identity. So each oil having their unique identity. So by using this technique, you can do uh, this analysis. Then determination of aflatoxin B1, B2, G1, G2 you can do in the food stuff by using the Hptlc. As we all know, being fluorescent, most mycotoxins are easily detected by Hptlc, and in the ppb and ppt level we can quantify that. Here no derivatization was needed. If no other fluorescent substances are present, cleanup not required. So here you can see the aflatoxin B1, B2 and G1, G2. Detection of sugars. We all know that monosaccharides are very difficult to separate by any chromatographic technique. But this Hptlc technique giving the very fast, cost-effective method separation, and you can detect the sugars with a very simple and low cost. So here you can see the glucose, sucrose, maltose and fructose can detect by using this technique. For this derivatization was needed, and thus derivatization was did by the ADP, aniline diphenylamine phosphoric acid reagent. So you can detect this sugar by using the Hptlc. Then this is another example of the nicotine in tobacco, gutka and mawa and pan masala sample. So it is visible in the UV also. But after the derivatization with the dragon drops, you can see how the prominent and intense band was observed. Then detection and quantification of water soluble and insoluble dyes. We all know that the Sudan dyes are banned because it is carcinogenic. So these dyes are not permissible in the food material, like in the chili powder they are adding to giving the good reddish color. But if there is added, you can do this and detect and quantify these dyes. Then there is a adulteration of toxic argemon in mustard oil sample also. So this we all know sanguinarine is a toxic polygonal ammonium ion present in the arginine oil, and this can be detected. And if there is any adulteration was added, so that also can be detected by using HPTLC. Detection of the palm oil in the edible oil. We all know that palm oil is little bit cheaper than the edible oils. So sometimes what happens, they added into that edibles oils, and if you go to identify or detection, you can go by this technique of HPTLC. So this is an example. So palm oil separation was here. You can see these bands, and there is a different oil: coconut, mustard, olive oil. And if they are also showing the like same pattern, or these all are present in this oil, so you can say that this is also adulterated with the palm oil. Then identification and quantification of the monosodium glutamates, MSG. We all know that before 7 8 years ago there was Maggie was banned because of this MSG, monosodium glutamate, which is a flavor enhancer commonly added to the product. And this is not good after certain limits, so this is banned. uh, which is used in the noodles, pasta and raw materials, that also we can identify or detect by using this HPTLC technique. Then this is one example of the new application field of instrumentation, the ink project in the US Security Service. So in the foreign countries, in the US, there is a specific ink was used for the legal documents. If any another fraud documents, forging, if happen there, so you can identify that also by using the HPTLC. Now for the HPTLC analysis and for literature study, there is a lots of books are available and online sites are also available. So these are the books you can refer for plant and drug analysis. Wagner book, then iCraxer book was there, ICMR books was there, and there is an application HPTLC Association. So this is the very free website. If you go on to the Google, you just type HPTLC Association. So this site will be open. You, if you register on this site by using the login ID and password, you will get a lots of validated methods related to the HPTLC analysis. So this is one example of HPTLC Association method. Then this is the she is the Dr. Vandana Gawande madam. I shown you one example of the degradation of cefixime and azithromycin. She did that analysis, and for that analysis she won that reward of Euro 2000 scholarship. So most promising researcher at Berlin HPTLC symposium. So for that analysis she used our HPTLC technique, and she got that award. Then this is the gold prize winning poster of HPTLC Symposium, which was held in the Bangkok in 19 November 2018. So Yugendra Patil and research guide Dr. Sunita Shailaja, which is the Ruya Autonomous College, ma'am, was there. Then the Anchrom having the philosophy, Anchrom wants to be your analytical partner, not merely the supplier. That means if you are taking our instrument, We are not only supplying you, but we will keep you updated and productive for years due to our excellent service, laboratory, and focus onto the customer. That means we are technologists, not traders. कि भाई बेच दिया हमारा काम हो गया, ऐसे नहीं है. उसके बाद जब भी कस्टमर को जरूरत है, when you are needed, that time we are always there for you. So this is our philosophy. We are technologists, not traders. This is our newly expanded lab. We unlimited support to the customer. Hptlc is accessible to one and all Indians. All common software and instruments for training and our analysis. Then there is a very separate set of instrument for hands-on training. You can see that. Then PhD earn and learn scheme was there also. Lab available for your publication works. If you are doing any good analysis with by using the Hptlc, you are always welcome to our lab. Lab is available for your publication work. Just like that Vandana government won that scholarship award. Then Hptlc education webinars with the live demo. We are arranging. Then, as I earlier told you, free training at Anchrom for government labs and PG students, postgraduate students. Training duration, the flexible times we will or mutually convenient rate at their places also. After the each training will provide the certificate. Till date, 5,000 scientists was trained on the HPTLC, and we have the MOUs with the several prestigious universities. And for that, you can email us on the hptlc@ancrom.in or the lab@ancrom.in, and or you can call us or WhatsApp us on this number. This is the Anchrom YouTube channel. So here you will get the data, lots of videos related to the HPTLC analysis. Anchrom aims to make India as a world leader in use of HPTLC. Our lab gives access to the all Indian who wish to use HPTLC, and but if they don't have it. So this is the Anchrom's aim, and thank you from whole Anchrom team. And I completed my PPT presentations. Now we will see any Q&A session was there.
Yeah, Amit, thank you so much. Now we'll have a small video briefing of what HPTLC is there, and then we'll move on to the QA session. I'm sure we have some additional time, sir. Can we
Yes,
extend the
yes.
session? Yeah.
Yeah, sure, please.
Yeah.
We will have few questions as well from the participants.
Yes, sure, sir.
Sure.
So before starting that, I'll just like to do a video briefing of what the entire process is. So thank you,
Sure.
Amit, for this wonderful insight of HPTLC. And I am Dr. Saikat, and I'll be showing you one of the analysis videos which are there. Unfortunately, we conduct this live from our laboratory, but due to this COVID conditions, we are restricted to show you from whatever videos we have as a resource. So definitely what I'm going to show you will be giving you a lot of clarity. So as Amit highlighted that the HPTLC process can cater to a lot of your daily to daily analysis, and the main advantage is it is quite flexible and it does not require any preparation time. You are any time ready to go about and you can start your analysis, and you can do a lot many things, not just plain analysis. And the best part is a single set of instruments does all kind of jobs, so you don't have to have lot many instrumentation like you have in other fields. Maybe one detector for this, one detector for that. Not like that. A single instrumentation is capable to handle all kind of analysis which you require in. So let me start you by showing a small video briefing about stevial glycosides. I'm sure you must be knowing about this plant of stevia. If not, then let me explain you in a very short description that we all know that artificial sweeteners are there for all of us. We are all getting fat, and then we all want to have slim figures. So we eliminate using sugar. And there is a new concept of artificial sweeteners. So among them, there are many sweeteners in the market like aspartame, saccharin, and they all have their advantages and disadvantages. More or less, the disadvantages are more. But one such molecule is Stevial Glycosides, which is obtained from the plant of stevia, which is a natural plant, and the Stevial Glycoside is also obtained from this plant, making it a very safer option amongst all the artificial sweeteners. It makes your daily to daily sweeter items very interesting to have with very smaller concentrations. It is around 200 to 300 times sweeter than the normal herbal artificial sweeteners or a normal sugar. So that is what makes it that even pinch of these sweeteners make your recipes and your food commodities, and you have a whole lot of stuff which are sugar free, we term it as, and many diabetic patients suffer, they cannot have these sweets, but with this artificial sweetener they can definitely consume sweets with the pleasure of having sweetness. So today we'll just see a briefing video of the entire analysis. Now today's video, what will be showing you is about one such formulation, the jam, which is prepared by this stevia, and it is being used for regular consumption. And what is the important aspect is we want to find out whether the concentration claimed by the company is right, and how it is done. That there are two approaches: either you have the pure extract of Stevial glycosides and glycoside and you compare it against your formulation, or the another way is to have some markers. So if you see, if I tell you that I have a maybe anti-headache tablet, and in that one of the components is paracetamol, okay, so it may be a combination of 2 or 3 drugs like acetaminophen or diclofenac along with paracetamol as one of the components, and out of the all I am interested in the the paracetamol. So paracetamol becomes my API of desired interest, it becomes my marker for assessment. And the same approach is in stevia glycosides is a composite mixture of 3-4 compounds which are there, and out of that one component is your stevioside A, stevioside B, stevioside D, and also you have ribaudioside. So this experiment talks about seeing those 4 markers simultaneously estimating into that formulation and correlating it with the presence of it in the formulation. So the very first process is starting off the preparation of the sample. So as you know that Hptlc is done on a thin layer chromatographic plates, which are there, thin layer plates. So these plates are disposable. So we have a stepwise approach when we are doing Hptlc. The first step is preparation of sample. Second is preparation of the analysis. Sample application are the steps of analysis which we do. Application, development, derivatization, documentation, densitometry, quantification, and followed by that MS validation of the entire study. So it is done in stepwise approach. Now even if this stepwise approach I am doing, my colleague can simultaneously do another experiment, and he may be at a different step. He may be at the development step, I may be at sample preparation, my other colleague may be doing application. So multitude in a single unit time, many of us, the same set of instrument we are working. As you saw, first what has been done is that sample has been taken, that is the jam. You sonicate it 15 minutes after adding this particular organic solvents. Methanol and ethanol are most recommended. Excuse me, I'm extremely sorry about that. But when we come back, we have this sample. Normally you can filter this, or we have an alternative methodology of centrifugation. When we centrifuge it, we get a clear supernatant which can be directly used for your analysis. The solvent is taken such that it can dissolve your components. For example, if I have the jam, I don't want the other sugars maybe interfering, or the gelatin part which is water soluble, I don't want their interference to come in. So instead of that, what I can do is I can take an ethanol or methanol and add into that sample, so that only those ribaudioside and whatever the organic compound which I want to extract it, that I can extract. So after doing that, the next step, as I told you, we program the software with all the parameters which are required. This is the application step where we define from which rack are we doing the application. So application is done band wise with the ATS4. Now in this ATS4, what happens is samples are taken up in the vials. I will show you the briefing of how this instrument looks like. But let us see that this is the process of application which happens. After the application process is completed, the next step, as I showed you, it is the development. So before that, you have to prepare the instrument to do the performance, and this is happening over here. You add the solvent into the ADC2 and you place the plate inside. Once that plate has been placed, the development of the on the plate of the mobile phase, which is actually moving onto the surface of the plate by diffusion. So how this diffusion works is that you may have one end of a cotton cloth, okay, and you just dip it in water and you leave that other end open and dry. Gradually what will happen, the water will seep into the cotton, and due to capillary action it will be wetting the entire cloth. And this is the same principle when you place the plate, it is wetting, the entire surface is getting wet, and the solvent is actually interacting with the silica and your analyte. So depending on its likeness to dissolve the sample, it will move the sample onto the stationary phase, and it will immobilize the sample at a particular distance when it reaches at a particular distance. Unlike other types of chromatography where the sample actually totally eludes with the solvent and it is traveling along with the solvent, here it is traveling along the solvent, but the silica is resisting it from passing by, and that is why at a certain point of time, when it reaches a particular point, the solvent, the entire amount is gradually diffused to a certain extent where it is blocked at somewhere in the center or at the top or at the bottom, depending upon the solubility of the substance. And that is how the separation happens on the TLC silica base. So this process is done totally automated. The next step is doing the derivatization process. So we have this immersion device and then another spray derivatizer which is there, which actually sprays the reagent onto the top. This is the dipping technique where you dip it, and instead of having this chemical, so what is this process is, in this process, inside the chemical, the plate is dipped in, it is removed, and then an external source of energy is given, such as heat, which accelerates the chemical reaction and turns non-visible compounds to a visible state. So right now you see that there are no bands available seen onto the plate, but they are present onto the plate. After the derivatization, after the dipping, what happens is these molecules, after heating, start getting visualized. So heating has to be done properly, uniformly, so that all the plate gets the same quantity of heat, so that the reaction is uniform throughout the plate and you visualize the plate. Very importantly, also the time has to be noted, how much time you are heating. Once you've done that, you get the plate and you start taking the images. This is done through an imaging instrument, the visualizer, and this is also done through the software. So you have a small physical process related to the actual performance of the step, and a larger software program step which happens automated, which is done automatically. So here you do take the images of the plate and store up in your file. Once you store this in your file, the next step comes to do the quantitative estimation, that is done through the scanner. So this is how the image has been taken. And now if you see that we are now moving into the next step, that is scanning of the plate. In the scanning of the plate, you convert, actually with the help of a technology, you are actually converting this visual information into peaks, that is the chromatogram peak. Now the chromatogram peak is a directly response of the molecule placed onto the plate. So you have different fractions separated. So each fraction will have a selective peak which is being generated by this instrument known as the scanner 4. It has got a huge range of wavelengths through which it can scan through, and each particular wavelength it will give a chromatogram of the molecule if the molecule is responding to that wavelength. So what happens is, after the scanning starts, everything internal adjustment is done automatically, and then you start generating the chromatograms. You can see this is how it looks. Now in this case, we always play with the RF value, that is retardation factor or representative front, many names it is known as. But the importance is in this, the RF value or the position is important. There is no factor of time involved over here, like other types of chromatography where RT is retention time is seen. And this is how you see the 3D view. So what you saw on the plate, that has been scanned, and you get a 3D plot of this the chromatogram known as the chromatogram has a composition of peaks which represent each section separated on selective tracks of the plate. So after substances, you have to assign this substances. So like here you see ribaudioside A, B and D, and stevioside is also there. I'm sorry, I had earlier mentioned vice versa, but it is like this. And finally you generate the report over here with the linearity calculations and everything into it. So it does not end over here. You have a TLC-MS interface, which is a kind of hyphenation. So existing Hptlc, you can hyphenate it to get either MS data, or you can also collect it in vials and do further investigation of that molecules. That is also possible. So here you see from the plate directly the spot is being picked, and then it is sent into the mass. So this is a TLC-MS interface where you place the plate, the spot is being eluted, a solvent is pumped into this TLC-MS interface, it is eluting the solvent and going into the mass. When it goes into the mass, it will start generating the mass spectra, that is another instrument, the mass spectrometry, which is also known as MS. In that you get the mass spectra. This looks like here, and so you can actually analyze a number of standards which are there, ribaudioside A, B and C. Of course this is a separate subject and not a part of Hptlc, but such kind of hyphenation is also present. So this is a gist of how the Hptlc process happens. Now within another 2-3 minutes I'll show you individual steps also. So first is the ATS4, which I was talking about. So this is the applicator of Camac.
The instrument parameter or rapid when working with to automatic.
Okay, I'll show you the physical
With its
part.
guided
So this is
approach, the
ATS4,
VisionCat's
which it looks like.
Hptlc
And what we do
software
is we place this
effectively
stationary
leads you
phase
through the
onto
entire
the
chromatographic
edge where it is
process,
being loaded.
step by step. Position the TLC Hptlc plate on the self-adjusting object support.
Okay, and this is your sample.
After having loaded the ATS4, you can start
you
the
have
fully
the wash
automatic
bottles
sample
on the
application.
top, which rinses the
VisionCats
syringe after
displays
each
the current
application.
status of the sample application and the expected end time.
So this is
An
how
application
process
sequence
of,
starts by
syringe
rinsing the syringe.
fill-up happens.
Next, the septum is punched and the needle lowered into the sample vial, thus allowing the syringe to be filled with the sample solution. The septum punch is retracted prior to the needle being removed to ensure the outside of the needle is wiped off by the tightly closing septum. The syringe moves to the programmed position of the plate and starts applying the desired sample volume. Several applications of the same sample are applied in sequence. Samples are applied as bands by default. Spots and rectangles can be applied as well.
Spots and bands. And once all the application is
Once
done,
all samples are
you can
applied
do
successfully,
further things after
just
that.
open the cover and remove the applied
Now you
Hptlc
remove the
plate
plate and
for
you are ready to go
chromatogram
for
development,
the next step.
the next step of the Hptlc process.
Now you have the developing aspect, how the development happens. Let's just see that. Again, there's the
Distance.
software part
VisionCats
which we
effectively
earlier
leaving
saw,
the analyst
and this
free
is how
to
we start for the
do
physical
automatically
process. So
through the entire
here it
chromatographic
starts.
process,
You have
step
these
by step.
parts of the Hptlc system.
The ADC2 will automatically develop the plate,
So you
leaving the
place the
analyst
filter
free
paper.
to do other work.
It's a Wattman filter paper,
The
which is being
ADC2
kept
provides
on the
development
backside to
under
saturate
controlled
the chamber
conditions.
with mobile
Open the
phase
front door,
vapors for
load the applied plate
equilibrium.
in the plate holder and insert the plate
It is
into
not
the
affected
automatic
by
developing
environmental
chamber.
parameters. Then you put your plate in such way into the ADC2, and this entire process after
Pour in 10 milliliters of the developing
pouring
solvent
the
for plate development
mobile
and
phase,
25
you go
milliliters
in
of
for the
developing solvent for chamber saturation into the solvent
command of
ports.
executing the step.
The entire process is monitored. The plate is developed in a saturated chamber if using the default settings. The automatic developing chamber ADC2 and the optional humidity control
On the right hand
create
side,
standardized
humidity
conditions,
control,
obtaining
which actually is
reproducible
responsible for
results
getting
from plate to
reproducible
plate.
results. You can see the
Perform
mechanism, how the
chromatography
air
under different
flows
climatic
and saturates
conditions
the
and
chamber
compare chromatograms created in different labs
and
of
removes the
results
excess
obtained
moisture
from
which is
different
there
batches.
and conditions the
Chromatography is
chamber with
started by
the
lowering the
dedicated
plate into
moisture,
the
and the
developing
plate also
solvent.
removes the excess
During
moisture, and this is how the
chromatography, the
separation
position of
happens
the
on the
solvent
plate,
front is monitored. At
and
the
this is
end,
being
the
monitored
developed
by the
plate is
software
homogeneously dried in an airstream.
automatically, and it is
All
being
parameters are recorded in compliance with
recorded.
CGMP, CGLP.
So this is how the development happens. And finally, let me show you a glimpse of the spraying, which will be the final step. Scanner, we have already seen, it's not much of a thing which can be described in, but let us see the step of
10 centimeters. A
this
small
is the
hood.
spray on technique of the latest one.
Select the
You have
small
seen the
hood for
immersion part. This
Hptlc
is about
plates
the
and the appropriate
spray
nozzle.
on technique. So
To
you
meet the
have this
diverging
hood, which
physico-chemical
actually seals
properties
the
of the
fume
different
from coming out
reagents,
from this
4
process
different
of
color-coded
derivatization.
spray nozzles are
Generally,
employed,
spray
and the user can
releases the
select from
fumes
6
outside,
spraying
but here you
levels.
have in the dipping also you saw it is environmental friendly. It does not lead to
Recommended
any fume
settings and
exposure, but here
recipes
also
are
it is being
provided.
contained with the help of this enclosed chamber. The plate stationary phase is
After
placed
positioning
inside.
the plate, the hood is lowered and completely sealed to prevent spraying reagent from leaking to the outside. Fill the
What you do is
recommended
just
volume
add
of
2 ml of
reagent
your
into the
derivatizing
nozzle.
reagent.
The
In
spraying
dipping technique,
process
you
is
require
started by
around 200
selecting the
ml,
spraying level
whereas
and
here just 2
activating
ml
the
of
start
reagent
button.
is required for a smaller plate. And you can
The
see
nozzle
how
generates
fine
an
mist
extremely
of
fine
this
reagent mist,
derivatizing
which
reagent
evenly
is being
distributes in the
sprayed,
hood and
because of
gradually
which there is no
condenses on the
diffusion
plate.
and the bands are absolutely sharp and quantifiable even after derivatization.
Reagent remaining in the gas phase is automatically aspirated and
So
collected in the
you
wash
see the
bottle.
excess fumes are
When the spraying
cleared with
process
that
is completed,
solvent deposition
push the
over
lift
there in the
button
bottle
and remove the
and
plate for
lower
further
the hood,
processing.
open the hood and remove this plate.
Visual observation of the plate
You can
shows
see 2
very
plates
high
which are being done
homogeneity.
using the same, show exactly
Reproducible
the
and
same kind of
user-independent
separation. There is
results are
no
obtained from
deviation. So this is about the derivatization process. So these are the critical processes of HPTL which are actually very important. Now I'm sure Amit had shown, just let Amit verify. Hello Amit, have you shown the HPTLC Pro
Yes, sir.
video?
I shown them
Okay,
that
sure.
video. Akshay sir
Okay,
explaining.
so this is all about HPTLC. I hope this has given you a clear-cut idea of how the process of HPTLC is being done, right from sample preparation to the application. Then we have the development, the scanning, the TLCMS part, everything we have covered with a very briefing, so making you understand how this process is. So with this, I hand over to you, Amit, and you can hand over it to Madan sir, who can start the process of question answer.
Okay, sir. Thank you so much.
Thank you so much, Dr. Saikat and Amit. It was rather a very wonderful session, and you have given us a complete picture of how the entire process goes on in the industry. And I hope students would have got benefited out of it. I now request students to unmute yourself or to unmute, and you can interact with them for any questions or clarifications that you have. Mr. Amit, can they unmute themselves, or do we have to give them the permission? I think
So they can unmute themselves. Those 4 students who have raised their hands, I have given them permission to unmute.
Bhargavi, if you have anything to ask, you can ask because you are there in the panelist room.
Yes, sir. So I have a doubt based on quantitative analysis. If we are not available with, is there any possibility to analyze the sample in
So,
nanogram or microgram level?
see, this is a question It's very interesting. The question of quantifying without the absence of standard. Now, there are a few things which need to be understood when we go for quantification. The response of the detector with a known quantity. Okay? Whenever we are interested in finding out some unknown or the quantification part, we need to understand what is the response of the detector to a particular component which we are analyzing it or which we are interested in. In absence of such relation, it is very difficult to understand that what unknown component is or how much it is generating. Even if you say impurity, you always have a correlation to the factor. You see any format, but you will require something as a reference to come to a conclusion. So if not, you don't have a very specific marker to the plant, but a general specific marker like beta cetosterol, which are very cheap, caffeine, these are very cheap markers which are available. But at least they can tell you if you want to correlate it and establish a relation. Based on that, you can suggest a quantification. But for the best quantification part, you require the standard, that is must. If not, these alternative approaches can be utilized. But definitely when you, in terms of quantification, you say you will require a standard. And that is what expected. In Hptlc, we have a provision of comparison viewer where we store the data which has been done over the past time. This is in three formats: the image part, the chromatograms, and the spectra. So these three information which generated from the sample, it can be stored separately, and you can prepare your own library, and that can be accessed. So in that case, you may have a standard at the time of investigation, but one time purchase you do, you prepare that analysis and keep it in stock. And whenever you require, you don't need to buy, you just compare your file with the same mobile phase you have to develop. After developing, you just compare that file in the comparison viewer, and from that you can get an estimation of what is the quantity. So that is possible, but marker is an essential commodity when you want to establish a quantity estimation.
So for example, like if I have quercetin, colchicine, only that standard or that sample can be standardized, can be quantitatively standardized in my sample, right? I cannot go for any other, even though if I have standard. Any other
I'm
samples
really
cannot.
sorry, but I'm missing your question. Can you just repeat, ma'am?
So for example, if I need to analyze alkaloids in my sample,
Okay.
I may have many number of alkaloid samples that can be tracked in
Absolutely.
my tracks or in my bands.
Yes.
If I am only having one standard, only that standard can be nanogram level can be seen from my sample also. No, I cannot get all the bands standard, even though
See,
if I'm
you
having
can
standard.
establish a correlation. See, what is that? See, I will explain you how this correlation works. Suppose you are getting 3 alkaloids from your particular band, and out of that, you know you are having one alkaloid substance, and from that you want to establish all the other 2 alkaloids which are there. First of all, the part will be like the other 3 alkaloids in intensity wise or and in chromatogram wise, where you can generate the peak area. You can estimate that it is of what percentage. So all the 3, how much percentage collectively they will be making 100%, but individually how they much they are contributing. Suppose this is my middle peak which I have a standard, okay, and for that I'm getting an area of say 1000, which is around 50% of compared to other 2. This is 25, this is 25, and this is 50. So if I have a standard, I can, and I know this value, then based on this, I can make an assumption that these 2 will be in this much quantity. But there is also a factor known as the detector response. Every molecule has a varied detector response. It may not be linear, it may not be exactly what you expect, that it may at times be exponential. But at least when you use this relative technique, you will be in a position to comment that okay, these 3 markers collectively making 100%. So if this is my 50 microgram and this is 25, so it should be around 25 microgram. So total it is 100 microgram altogether, which I have. This estimation you can do, and you can get to a preliminary understanding. This is also approach is used in many of the impurity profiling, where a related substance is being used to generate the known impurity, which is we don't know how much it is there. So we relate it to the known compound, and then we establish a correlation, and then get to understanding that, okay, when I have 100 grams of my sample, this impurity represents 0.1% or 0.2%. So like that, it is possible, and you can investigate, definitely.
Thank you, sir.
There is one question in the chat box. Is
So
there any
then-
possibility for analyzing protein samples? I have seen only one or two literature.
Very good. So proteins from last webinars which we have been doing, last I think so 8-9 webinars which we have been doing, this question has been coming up to us. A lot of people want to do protein estimation, and they really want to investigate into this. So let me clarify this. Protein is, molecular weight. This concept must be known by everybody. There is attached to each molecule, there is a weight attached to it, molecular weight it is termed that. For example, hydrogen has 2 molecular weight, oxygen has 16 molecular weight, carbon has 12 molecular weight. That is how it works out. So every molecule, when you say a protein, it is a complex molecule, and it is generally in kilodaltons the molecular weight runs to. And what happens is this TLC plate, it has a limited capability to handle molecules of very large chains. So if you have a very high molecule which is there, which is passing through that silica, it will not be able to resolve it because of various factors involved in it. And that is why you have specialized proteomics plates which are available, which can handle 25 kilodaltons maximum. And in this also, you can use a technique known as hydrolysis of the protein, and then you can analyze this hydrolysis of the protein, breaking it into amino acids, and that can be analyzed, because amino acids are possible on Hptlc. But if you say that you have an entire protein, that entire protein will be maybe possible with proteomics plate, but not with the normal silica plate. In that case, you have to fragment that big chain into smaller parts by hydrolysis, and then analyze breakage patterns which are there into your analysis. So when you are doing an experiment of hydrolysis, do be careful how much amount of solvent you are putting in, how much time you are exposing it for the hydrolysis, because these all things matter very much. Even the minutest of the details are very important to know when you want a reproducible result. Otherwise, what will happen, if one sample you put 2 ml of hydrochloric acid for breaking the protein, and in the other one you are adding say 3 ml, so that may lead to changes in the sample preparation. So the sample preparation has to be uniform. But proteins can be analyzed onto Hptlc system, but with a limited capacity, but definitely it is there. And we also have an alternative instrument which is not a part of this session, that is in Hptlc. It is known as the DBS 100, that is dry blood spotting, that is DBS it is known as, and it is directly hyphenated to the mass. An extraction is, you have to just you have card like structures or filter paper kind of structures. On that you load your protein molecule. In this case it becomes blood, but you can use some other molecule, and that is being directly extracted and sent into the mass. And you can, if you have an HRMS for higher molecules which are there, at the time of flight technology in mass spectrometry can handle such protein analysis. You can find out the protein from that. So that can be an alternative over here. Hptlc can do, but in a limited way. You will have to do some research work before you actually want to do some experimentation, because as we are doing it on laboratory, we don't handle protein samples because of our working atmosphere. We are in a restricted residential area and have limitations of what biological molecules we can handle. We cannot use too much of solvents and things like that, because as I told you, these are the factors. So that is why we have to take care that we are kind of a showroom, but at the same time we also give you some experience. It is not that it's just a showroom kind of thing. So in that way I can just help you out in your protein request. Hope this answer would give you some enlightening of how you can proceed with.
Do we have any other questions? I could see Amrin had raised hands. Reshma, do you have any questions to ask? Amrin or Reshma, do you have anything to ask? There's something on chat.
Let's see. Okay. You have something written over here. I question for illusion of separated zones. Can you? Yeah. So somebody has asked the question. Let me see who it is from. There's a question for illusion of separated zones. You can use TLC-MS interface. Okay. I think so that is being answered.
Sir, all the questions are answered, sir.
These questions are answered. All of them.
Okay, great. So if any of you have any of questions coming in later, because science is always happening in your mind and research is starting from there, and sometime you come up with a question which you require some clarity, do write us at lab@anchrom.in. And with this, I would now hand over the session back to Madan sir. From the entire Anchrom team, thank you for this time of yours, which is very valuable for all of us, so that we use the time in a very valuable way. Hope this session was very informative, and we look forward to also interact with you in future also.
Yes.
So with this note, I will just hand over the session to Madan sir. Sir, over to you.
Yeah, thank you, Dr. Saikat, Dr. Amit, and your team. It was really nice because I know this technique is quite new to most of us in our institution. Though myself and Bhargavi, we have been using this, and my relationship with Anchrom is quite old because I know you since 2000. Myself and one more Dr. Kavita, we had been there about at least 15 years ago. So we visited Anchrom and we were there for some short training and things like that. But I lost contact after some time. So it's nice to get back. And it's very nice to see Anchrom doing a lot of things for molecule identification and separation. Yeah, thank you so much. May I now request Bhargavi, if you can just switch on your videos, we can take a print screen. Mr. Rahul and Bhargavi can also switch on your video, please. Thank you. Thank you. I now request Dr. Bhargavi to give the vote of thanks formally and to conclude the session.
Thank you, sir. I, on behalf of KONGUNADU Arts and Science College and the entire team, I deem it a great honor and privilege to propose the vote of thanks on this memorable occasion. First and foremost, I thank our chief guest, Mr. Amit Palande, the application chemist of Anchrom Enterprises. A heartfelt thanks to him and his valuable Hptlc insight, application, visualization, and the different modes of scanning methods and their evolution from TLC methods. It was valuable in terms of automatic analysis for the biological products for analyzing pure and crude sample variation in terms of bands comparing with the standards. And a heartfelt thanks to Dr. Sir for his informative HPTLC video and explanation for all the questions being answered by our invitees and the students. We are grateful to our principal sir for his words of encouragement, his able guidance, which was always encouraged. I thank him for his kindness, his interest, and his continuous support. I thank our secretary ma'am for her unstinted support for this function, her continuous hospitality, and her continuous support towards student-based education events. My heartfelt thanks to Head of the Department and Research Head PG Madan Shankar sir for organizing a valuable industrial session, his guidance and encouragement in all our efforts, and also UG Head Shivaprakasam sir, and also for all the teaching staffs who have worked hard to ensure that this occasion becomes a memorable success. And all the students, invitees present here. Last but not the least, I thank you, my friends, for your cooperation in making this event a resounding success. Once again, I thank you all for your attention. Thank you.
Thank you, Bhargavi. Thank you so much.
Thank you.
So let's hope that Bombay becomes all right, and maybe I would like to come see you all there. And also, when things are normal, I invite you all to our campus. I should tell a few things about our campus. This institution was started in the year 1973, and this was primarily started to serve the farming community. And I'm proud to say that we have got the DST FIST program for the second time now, for a tune of rupees 1.5 crores. We are also a Star College recognized institution. We have been recognized for 4 departments, and recently we also got the UN award for the plastic free campus. So this is just a bird's eye view of this institution. And we are very strong in research, and I should say our overall Scopus H index is about 43. So most of us are actually using HPTLC in our institution. And I feel like a lot of us will contact you in future for a lot of analysis, and also we'll be very much happy if we can sign a formal MOU with you and see how we can share and also give inputs to the industry as well from the little of research that we do in our institution. Thank you so much for being with us this afternoon. Thank you.
Sure, sir. Privilege is ours, sir. Thank you.
Thank you so
Definitely
much.
we'll meet. So with this, I request Rahul, if you can ask sir's permission and we can end this session.
Sure. Yes. Yes. Thank you so much.
If I end the session?
Yes. Thank you. Thank you.
Thanks a lot, sir.